So, bonjour, welcome everyone. It's a pleasure to have you all here for this panel session. So, my name is Diane Iglesias, and I'm a Director of Innovation and Programs at Genome Quebec. And I just want to make a quick announcement, which is that the Public Health Agency of Canada is developing its strategy for environmental surveillance of antimicrobial resistance. So if you feel you can contribute with your expertise, please, I invite you to use this QR code and enter whatever information you can enter there that will help the Public Health Agency of Canada. So what I will do is to invite each of you to present in 2 minutes your research. So I will start with you, Emilie Bédard. She's a professor in civil engineering at Polytechnique, Montréal. She studies and conducts field studies on environmental microbiology, engineered water systems, and opportunistic pathogens.
Thank you. Does that work? Yes? Okay, thank you. So thank you. Good morning, or well, it's almost lunchtime. Hello, everyone. I'm thrilled to be here today. My main research focuses on water microbial quality, on the urban water cycle and built environment. And today I just want to touch on 3 projects that are really related to antimicrobial resistance. So the first one is tracking AMR through the urban water cycle. So it's really touching every step of the water cycle. And Helena, a PhD student, has been working on that project, doing field samplings in several locations throughout 2 cities, and also conducting a survey to understand people's perception and habits on AMR, and looking at the correlation between antibiotic usage and ARGs that are detected in the wastewater. And hopefully our results can help guide some efforts to limit the progression of AMR in geographical area of Quebec. And there will be a poster tomorrow on that. The second project is a project that's just starting now and what recently funded with the opportunity with Genome Quebec on controlling AMR bacteria in sink drains in hospitals, and really looking at the outside factors that are influencing the microbiome and the ARGs that are present in the sink drains in collaboration with 6 hospitals from the Montreal region. And the idea is to develop a toolbox that could be used by hospitals to really tailor their action on their controlling the AMR in their sink drains according to their specific characteristics. And finally, going further down into the cycle, as this gets all to the wastewater treatment plant, we're looking at the role of different steps of the process in promoting and dispersing AMR. So there's been a lot of talk about that in this conference, but we're looking at specific steps in the processes, how they contribute to the dispersion of the aerosols for opportunistic pathogens and ARGs, but also the impact of disinfection, UV, and ozone, to try to identify best operating practices and mitigation measures to limit ARG transfer, selection, and dispersion. And there will be also more information on that at the poster session tomorrow.
Great, thank you very much. Next would be Louis-Patrick Haraoui. He's a clinician researcher focusing on antimicrobial resistance throughout the One Health spectrum. He works as a physician at Hôpital Charlemagne and is associate professor in the Department of Microbiology and Infectious Diseases at Université de Sherbrooke. He's a Fonds de recherche du Québec en santé, clinical research scholar, a CFAR Azrieli Global Scholar, and holds uninterrupted research funding since 2018.
Thank you. I'd like to thank the organizers and Genome Quebec for organizing this panel and inviting me. So the best way to prevent the spread of antimicrobial resistance is to prevent the resistance genes that underlie the process of resistance from getting into human pathogens. And most resistance genes come from the environment. So what we want to do is try to screen the environment to prevent that. And many people who've been doing this have been doing some genomic sequencing and trying to see if there are resistance genes in the environment that look similar to what we know in the clinic. And I want to show you why genomics are useful, but they're a partly insufficient tool, and how combining it with structural biology and artificial intelligence can actually improve our ways of detecting new resistance genes before they make it into the clinic. So I'm taking as an example a very common resistance gene that some of you may know called NDM, which is a carbapenemase, a very problematic resistance gene on a worldwide basis. It was discovered about 15 years ago. And when we compare NDM to other similar resistance genes, what we see, for instance, when we compare it to VIM, is that it only has 32% amino acid identity, meaning that its sequence is very dissimilar. So using approaches that only rely on sequencing are insufficient because most people will use higher cutoffs of 60 or even 80%. What happens if we actually use a tool like AlphaFold or other protein 3D prediction models to try to see what these resistance genes look like? So this is VIM on the left, and this is NDM. And if we superimpose them, they actually look very similar, but we can actually have a tool to tell us how similar they are, and this is called the template modeling score, which is a score that ranges from 0 to 1. The closer you are to 1, the closer the two proteins are. So here we see two very dissimilar protein, or genes rather, I should say, that are extremely similar once they're folded up in the protein. So this is what we do. This is one of the projects. This is the project that Genome Quebec has financed. We do environmental screening, or we use public databases that have a bunch of metagenomic sequencing. This is water. This can be permafrost. This can be many other sources, environmental. We convert the sequences into amino acids. We use AlphaFold. We look at the protein structure, and we determine the TM scores. And then what we do is we use these genes that lead to proteins with high TM scores, and we put them in bacteria in the lab, and we test them out. So this is work that a PhD student, Lachlan Bartrop, has been working on. And so far, we've essentially identified novel resistance genes to, this is very preliminary, but I'm sharing this with you today, 3 resistance genes to trimethoprim, 1 to gentamicin. We're working on potentially novel resistance gene to sulfamethoxazole and clindamycin. And we're working on improving our algorithm and pipeline to decrease false negatives, because obviously we don't, our ratio of pickup is not 1-to-1. So we'd like to get it as close to 100% as possible. So I'd like to thank you and just mention quickly that we have some graduate students positions open if people want to apply. Thank you.
Thank you, Louis-Patrick. So next in the line is Paul Thomassin. He's a professor at McGill University and an agricultural and resource economist. His research area includes sustainable agriculture, technological adoption, the evaluation of agriculture and environmental policy, the economics of healthy diets, and the burden of illness from foodborne pathogens. Quite busy, Paul, so the floor is yours.
Thank you. So, good morning, everyone, and thank Genome Quebec for inviting me to come to the panel. We had a project funded through Genome Quebec and the Public Health Agency of Canada, and basically what we wanted to do was build a bridge between sort of human exposure to antimicrobial resistance in terms of foodborne pathogens and estimate the burden of disease. Yareen Yu, my graduate student, gave a presentation yesterday on that, so the results are all there. But so this is sort of an overview. Again, our project itself, we had a large number of researchers, both in terms of the project team, but also in terms of collaborators. So it was a very integrated project that brought a lot of different views to the table. We are taking a One Health approach, and so if you look at the diagram on your right, or my right, what we're looking at is we're looking at sort of pathogens moving from sort of the farm through the processing sector to the retailing sector, and we're looking at exposure to AMR through that process. And from a scientific point of view, or from all of the microbiologists, they get all very excited about AMR and proteins and all that sort of stuff. But I'm an economist, and though that's all very exciting, it really doesn't mean a whole lot to me. What means something to me is that people get sick. And so what we're trying to do is to try and bring some of that science into a decision-making process. So where you could actually take the science and get results that are meaningful for decision makers. And one of the ways we do that is actually to estimate the burden of illness. Now, the burden of illness, so for example, if you get salmonella, and if you get AMR salmonella, maybe you just self-treat yourself and you get better, and one of your outcomes is going to be a recovery. But maybe you get sicker from that salmonella, and maybe you have to go see a doctor, and again, those increase the cost to our healthcare system. It also increases the cost to the individual. And again, maybe you get hospitalized, and maybe you recover, but maybe you end up getting sequela. So it may be a chronic disease, sort of something like IBS or RA. And so you're going to have a lifetime where you're going to have these added costs because you got AMR salmonella through the food system. And maybe you'll die. And those are all costs to society. So again, you have to remember the idea behind the economics is that we want to increase the well-being of society. And part of the ways we want to do that is to maybe sort of interject some policies within the food processing system, anywhere from on the farm through processing through the retailing, that we could actually decrease the amount of AMR salmonella or Campylobacter or whatever coming through the food system. So the idea behind it is we take the science, we actually estimate the costs, and then what we will do is actually look at different interventions to see how that impacts on the costs and the benefits. And so hopefully in terms of public or private health decision making, this becomes an important key in terms of looking at interventions within the system. So again, the idea behind this project is that, one, it takes a whole One Health approach going from sort of the farm all the way through the system. It's interdisciplinary. We have economists, epidemiologists, veterinarians all working within us. We have a partner groups, Public Health Agency of Canada, Guelph, and McGill, and we have different funding agencies involved. So the idea behind this is that we've now developed an annual and lifetime Markov chain salmonella and Campylobacter models that will actually predict what is the cost of foodborne illnesses coming through the food system in Canada. And the beauty behind this is we can estimate case numbers, we can look at the burden of illnesses, and also look at a quality adjusted life years estimate, so we can actually do both quantitative and qualitative assessments. The idea is to build this bridge between the impact assessment model and also the burden of disease. And basically what we want to do is look at further research to evaluate the cost implications of interventions in the supply chain. Thank you.
Thank you, Paul. Last but not least, Chan Mangat. He's head of the wastewater surveillance unit. His lab monitors SARS-CoV-2, influenza, RSV, Mpox, and now AMR across Canada. He's based out of the National Microbiology Laboratory in Winnipeg.
Thanks for having me. So the first thing I need to answer is the question that you guys posed to me, what's my genomics project? And that's designing a federal wastewater-based surveillance system for AMR. It's a tremendously onerous project, and we've just gotten started. We sort of foresee it happening in 3 phases, 3 technological advances, and we've just completed the first phase. And Jay Daigle provided a nice poster on day 1, that was poster 95 if you remembered it, outlining some of the things that we prioritized early on, which are really the quantification of antimicrobial resistance genes, and I think later phases were prioritized the contextualization of those genes, and then further quality control, let's say. All of this happened, not in a vacuum. So we're very lucky to work under the GRDI program. And so to plug somebody else's workshop, workshop number 8 is run by Jordan Broadbent and Dominique, and they'll probably, and I don't know the content, but you'll go over some of that collaborative work. And it's been beneficial, I think, especially in the context of the talk that you heard this morning from David Graham. There was a lot of discussion even before we got started. A lot of government agencies got together and said, we'll look in the environment for AMR. A lot of it had to be aligned to their own mandates. So it was sometimes tough to find commonalities, right? Like the Department of Fisheries and Oceans and Agriculture and Agri-Food Canada now have to work together. So that's not an easy thing to do. So beyond what you do, it's how you do it, right? And so there's some methods standardization that's going on. That's a hot topic at this conference. Maybe a little bit more boring, but very, very important is the management of data. That's being tackled as well. And for us, coming from the SARS world, it's been tremendously important to work in an open science environment. Whether that's internally with disease modelers and internal stakeholders, or just having that out to the public, right? Really all of this work, I should say, started prior to the pandemic for me. My background is in AMR. Wanted to get started with just 16 samples in the summer of 2020, and things have ballooned, right? So, the fortunate thing about all of that process is we've got lots of profile, lots of money, lots of excitement around wastewater. Maybe the more difficult thing is that people are asking tough questions very early on, I think, in the development cycle of this work. So I'll just go really quickly to what we've done. We started with a 9-week study in central Canada that included 2 urban, 1 rural, and 1 remote sites, so a small microcosm of the country. The thing that we noted is that remote sites were distinct, so here by Shannon diversity, and if beta diversity is ordinated in that way, they stand out, so rural and remote sites. And that's consequential because it informs what does a national program look like. There's less AMR in remote communities and rural communities. That means it might be a certain policy opportunity there versus what we see in the urban communities, which were essentially indistinguishable. And we'll look into this a little bit more, but at least over our study, wastewater is fairly stable, which means at least from an economic standpoint in urban communities, we might not have to sample so much. It's so expensive, especially I'm hearing about this high-C sequencing now, and we'll have to do long-read sequencing. The economics of getting that done, it won't be easy. And lastly is we have to build a surveillance system that's policy-aligned, right? So somebody wants us to look for H5N1 in the wastewater. Right now, the high-risk folks are people that work in the agri-food industry. Our network was built sort of bang for your buck, more people, and minimize the number of surveillance sites. That's because it was built for SARS. It wasn't necessarily built for AMR, but we're making some changes along that front. And I want to make one little plug for Ziggy. We're a major contributor here. PHAC, we have new and renewed interest in the environment, and Ziggy was super important for having us here in a big way. So thanks.
Great, Jan. Thank you so much. And I think you touched upon many different points that I wanted to bring to the conversation. As you know, and I mentioned before, Genome Quebec is all about democratization of genomics or omic technologies. And when we talk about democratization, we talk about adoption, integration, implementation. So I wanted to first start with the adoption piece. We know that as the threat of AMR resistance continues to grow, the traditional approaches that have been taken might not be as successful. And so cross-cutting approaches or technologies could help in the monitoring of emergent pathogens or antibiotic resistance. So I was wondering, in your line of work, how advanced is the adoption of genomic technologies in the study of AMR, and what are the benefits of integrating omic technologies in a One Health context? I don't know who wants to start. Emilie?
Well, so how advanced? That's a good question. I think that we're moving along toward that to include the genomics in the monitoring of AMR. But I think if we want to go further into this and have further increased adoption, I think we really need to streamline the protocols, the processes that even non-expert can start including that into their strategies to monitor and further their knowledge around AMR. So and accessible training. So I think those would be key aspects where we could go even further into adopting the genomics.
Anyone else?
I think if you view where genomics fits, I think I tend to see it as a very broad and long process because it goes from sampling all the way to actually analyzing the end results. And there's a lot of hurdles along the way. So there's one thing is just to get the ability to sample and to sequence or to do other things, but then it's how do you disregard the noise that comes from all of the things that come along the process and really are able to focus on what you're trying to do. So for instance, I have a project where we're sampling migratory birds and trying to see if there's any signs of resistance genes that are circulating within different populations with a kind of a gradient from rural to urban. But when you sample another living species, you're also sampling their DNA. And so when you sequence, you have a disproportion of bird DNA compared to what's really of interest to me, which is microbial DNA. So it's refining that technique. So yes, tools are important, and we need to increase the adoption. But then it's everything that comes along with it. How do you refine the use of the sampling, the use of the tools, the analysis that comes with it, to really minimize the steps and the work to get to the endpoint, which is what you're interested in.
No? Yeah?
Maximizing adoption, is that the question?
Yeah.
I mean, I work within like the public health laboratory network, I think, and raising the floor on capabilities is super important, mostly because we have to distribute this work, right? So, in a lot of ways, the NML has led some of the laboratory development, analytic development against genomics, and the capability is really good at the NML, right? So, lots of AMR is detected as part of, regular practice there by genomics, right? So I think most salmonella are tested, for instance, at the isolate level for genomics. The metagenomics, there's lots of pilot projects. There's no clear business use case. There's not, let's say, a standardized report that goes out where metagenomics is underpinning the report. But, as far as adoption goes, we've fairly good internal training programs to bring in new bioinformaticians and get people up to speed. It's just working within the provinces and territories and raising the floor there so that we can all take on sort of equally complex problems. And for lots of reasons, and for efficiency's sake, I think that'll need to get done, yeah.
Okay.
Just another comment in terms of the adoption. One of the things that we should be looking at is, with the adoption of the new technology, we're going to get better information. And the idea, I think, is that if we can get better information, then we can use that information to make better policies. So I think it's really important. It's interesting to get the new technologies and it's a lot of fun for the scientists to do that. But I think we have to look at the long-term goal is what is the information we need and how can we adopt those technologies to give us the information that is needed. And I think we have to think about it sort of from a social science perspective, from a either decision-making perspective or from society's perspective. This is the information they need in order for them to either change their behavioral patterns and or to have better policies.
Okay. And I wonder, in terms of integration, because I think that sometimes it's difficult to change things the way they have been working for a while, and even if there are new or better methods out there. And when we consider new surveillance methods, how do we ensure that they are well integrated with what is running today, with what is happening today? What can be done to facilitate this integration of new genomic resources and tools in surveillance programs? So, I don't know, Chant, maybe you're in charge of programs?
I don't know if I'm in charge. I wore a blazer today. I wore a blazer today, so it seems like I am. But I mean, I think for us, integration, it hasn't really happened in a big way, but we're trying to set the stage for a future program. I think data standards are probably the biggest thing that needs to get worked out across jurisdictions and hopefully to get some academic buy-in. I mean, one of the big reasons is because programs start and stop, right? Academic initiatives start and stop, and a lot of that data just gets lost, especially if it's not put in a place where it can be, understood after the fact, right? And I think it helps with buy-in, within a federated system, if everybody's operating on the same data standard, because then if I develop a tool or some sort of pipeline, then everybody can go and then use it. And especially with environmental surveillance, community surveillance, one of the things we're noting is that you have to be able to understand the community's composition. So its demographic composition, how the streets are laid out, where the water flows, right? And so you might not necessarily need that information yourself, but somebody else will need to do that and synthesize it with maybe your data stream. So having all of that in place is important to get the most out of the data that we're collecting right now. And it actually helps us decide where to go next, right? So for instance, for monkeypox surveillance, there's a certain at-risk community there, but we can't decide where to survey because we don't have certain contextual information about that at-risk community, right? So we have to go on other indicators, let's say, and it'll be the same for AMR and other diseases, yeah.
Okay. And how about you, Louis-Patrick, in terms of health, human health programs?
Yeah, I think as Paul was saying and others, that we generate a lot of data with all these new tools. The question is, how do you render them intelligible to different groups, different stakeholders who have different reasons for wanting to have access to parts of that data and how they will interpret it and use it. And that's a big challenge, So obviously, on the one hand, there's work on the surveillance and research community to try to reach out and understand what are some of the needs to be able to render them and translate them in a way that's accessible to the people who need them. I think also the people who want to have access to that data need to also work on their literacy. I mean, it's nice to almost spoon feed people, but you have to also be able to somehow reach people halfway in some cases. And it's important to improve people's microbial and microbiological literacy, I think, in general. I think, the COVID pandemic did a lot to help in many ways, but I think it's important to sort of raise the stakes on things like AMR because that's less well known among the public. And if we do that, I think people will more generally be able to reach a way where research and lay or clinicians are able to sort of speak in a more direct way.
Emilie?
Well, I wanted to add that I think to have better integration, we also need to think about what's before the genomics, because you can get results, but they're as good as what you've done to get those results. So adapting our sampling strategies to the processing required for genomics, I think it's really important. And also, if we want to share across different groups, different disciplines, we also need to standardize the way we do those sampling and treat samples, because otherwise you can't really compare or homogenize the information, harmonize the information between different sectors. So that's also something very important that I find when you read through lots of studies and papers, it varies so much and it's not always detailed enough to be able to interpret the results as a function of that.
Mm-hmm.
Well, I agree with Emily. I think surveillance is key here, and I think we all can agree on that. And I think Emily's right. We've got to deal with the sampling. From my perspective, some of the work we've done is that, to me, we have to also go to the other end of the scale too. Sort of like we have our Sentinel sites in Canada. I think we need more Sentinel sites in Canada. When we look at how we follow up when people get AMR, we do a survey, we follow up, but we could actually expand that to include, let's say, medical records. We're in Canada, we're now moving towards sort of electronic medical records. It'd be great if somebody gets a foodborne pathogen that is AMR. It'd be really great to follow up over time if they're going to get a chronic sequela what was the impact on them? How do we have to deal with this? That gives us better estimates of the cost and the burden on society of those types of problems. So I think surveillance is key. I hadn't thought about the sampling problem, but now we can see that it has to go all the way through from sampling to the techniques used, to the use of data, but also the follow-up on, in particular from a human health perspective, what has happened to the patient afterwards.
So I have two follow-up questions, and I can't decide which one I want to ask. But maybe just to follow up on this integration across different disciplines. So in emergency management, it is said that bridges are better built either pre or post-crisis. So what needs to be done now in order to be able to integrate all these different sectors, or human, animal, and environmental sector, to decrease this fragmentation, to increase the interoperability, and all these different elements that we have mentioned? Anyone has any ideas?
I can start,
Yes.
because it was talked about this morning when we looked at the graphs of how sectors were starting to integrate. There was, within the last 2 or 3 years, there was an increase in the integration between sectors. And one of the reasons behind that was presented was the incentives for projects to include all different disciplines. I think naturally people will tend to stay within their own area of expertise. So you really have to put forward programs that will, quote unquote, force the multidisciplinarity and the integration of the disciplines that don't typically work together or wouldn't think of it otherwise. So I think that's one way to facilitate or to promote that integration.
Any other thoughts?
I mean, I can speak from the perspective of trying to develop something new, which was wastewater surveillance, wastewater-based epidemiology. That's in the context of the Public Health Agency of Canada. There are lots of groups, that they're not all centrally organized, and they all have their own ideas about value and what should and shouldn't be done. And I think you need to develop your program. And go back to the idea of values, is that people have to see how it impacts their own mandates, their own goals, and if it makes their lives easier in some way, and that might not be, particularly what you survey, but how you survey it, the other infrastructure that you bring into play might make other folks' lives easier, and then you start to build a consortium, right? And then ideas can start to converge, data streams can start to converge, and maybe you can ask new questions that people couldn't ask before. And then I think, then you really get down the road of development and integration of your program. So that's been our experience. And underpinning all of that, at least internally, was being able to share data like nearly in real time with folks. Yeah.
So I think there are two. Well, I think I'll touch on the 2 points that Emilie and Chan just mentioned, but so increasing interdisciplinarity, yes. So I have a background in anthropology, and I'm a physician doing very kind of fundamental basic science. So I think trying to demonstrate to people how having different profiles and integrating them are important, and reaching out to people like Paul, who's an economist, and trying to bring in other aspects or dimensions is important. But I think there's also a lot of administrative and bureaucratic issues. And I'll give an example from my university, and then I'll give another example. But for, we have a microbiology department, which is in the medical faculty, and then there's a biology department, which is in the science faculty. They're on 2 different campuses, and there are people working on AMR on these 2 different campuses. And, we want to work together, but there's already all these minor obstacles just because we're in different faculties, we're on different sites, et cetera, and this is just within one university. So imagine scaling that up to much bigger dimensions. The other is that, there are brilliant government scientists like Chand, and most of the funding that I can access, I can't send money to Chand, and I can't have him on the grant with me because there's a lot of, there's these kind of ministerial or bureaucratic norms that prevent money from going from one place to the next. So it's great to promote interdisciplinarity, but we're often kind of also being blocked by a lot of administrative and bureaucratic obstacles as well.
I think Emily's right. I think, well, from a university perspective, money attracts people, and the idea behind it, if you get granting agencies that say you need interdisciplinary, then people have to seek out interdisciplinary. And there's two things. There's a lot of barriers, and Louis Patrick, you're absolutely correct. But the other thing that does come from that is that you start working with other people, and eventually relationships get formed. And what we see, at least from my experience, is that once you start working with other scientists, you start to see from their perspective how your work and their work can actually come together to address a problem that probably is better done than if one of you do it individually. So I think the idea behind it, there's got to be some incentives to get disciplines to work together. We try to minimize those barriers to those things, but once you start getting people working together, they tend to continue to do that over time, and I think that in the future will make for better academic performance and better policies and just better science.
Thank you very much. I don't know, 2 minutes? No? No. 2 minutes? Okay, no, I just was thinking if I would open the floor to the audience for just one comment or one question. And yes.
Jim Teege, Michigan State University. My question is with regard to culturing microbes. Your Genome Canada, or Genome Quebec, which is very genomics focused, I know some history about that as well, and this morning we heard from David Graham saying culturing is important. Is there a danger that we do not do enough culturing in the future? And I also mentioned, I heard from CDC about 5 years ago, where they used to receive isolates from state health agencies. Now that's down to less than 50% they receive isolates. They just receive genomes. So is there a danger, I guess, in not doing enough culturing?
So that question is addressed to me or the panel? Well, I can say that Genome Quebec as funders, we're not precluding any aspect to be included in the projects. So as long as there is a content, a genomics content in it, doesn't need to be necessarily 100% genomics. Could have components, chemical components, physical components, any social components, because we ask actually researchers to address LC or GELS questions. So it's not that we only fund genomics, pure genomics. What we really ask is that there's a genomics at the core, supported and surrounded by any other research that is requested or needed to be done around that theme. But I don't know if you recipients from Genome Quebec funding.
I could take it on partially just because I'm married to the person that does some of this stuff. But so a lot of antimicrobial susceptibilities done by genomics. It's tremendously cost effective, right? And so more and more pathogens, bacterial pathogens, are being brought online to that program, and it is an acknowledged risk that we could under phenotype some of our surveillance streams, and then we won't be able to ask questions of what's new and emerging, and it's being taken into account. So some fraction of isolates are being phenotyped at all times. I don't think it'll ever stop. And what's good is that the genomics a little bit guides you on what to do and what not to do. So your point's well taken, and it is being risk managed. Yeah.
Anyone else? No, you're good? Okay. So I think time's out, but I would like to thank our panel members for this very interesting discussion, and you, the audience, for participating here, and I wish you a good continuation of the conference. Thank you.